Next, I want to talk about the electron transport chain and ATP synthesis. So the overview of this is, there's a lot to look at here, and there's a lot of confusing numbers and molecules here. But just looking at it one piece at a time, ATP synthesis occurs in the inner membrane of the mitochondria. So inner mitochondrial membrane. And remember, there's an outer mitochondrial membrane, right? The outer mitochondrial membrane separates the intermembrane space and the outside of the mitochondria, so the rest of the cell. And then there's the intermembrane space, which is in between the two membranes. So think of it in between two walls. And then there's the inside of the mitochondria, which is this, the matrix, right? We're looking at right now the inner mitochondrial membrane. So that's the inner wall, okay? That's where ATP synthesis occurs. Part of the electron transport chain, there are 4 complexes, complex 1, 2, 3, and 4, shown here, each with their own purpose, as well as this big enzyme called ATP synthase, and it's very easy to remember. Synthase synthesizes ATP. It works based off both an electrical and chemical gradient, which powers the ATP synthase, as shown here with these hydrogen H+ molecules. There is a gradient of H+ in between the mitochondrial matrix and the intermembrane space. And this gradient is the basis of the power source, essentially, for ATP synthase. If you want to think of this as kind of like a motor, this is the electricity. This gradient will be the electricity that powers the motor. So we talked a lot about NADH and FADH and how they are equivalent to ATP. So how does that actually work here? Well, complex 1 and complex 2 of the electron transport chain use NADH and FADH respectively as a power source, which is why producing these reduced cofactors is so important. In fact, we've already actually learned about complex 2 in the last lecture in our about Krebs cycle. Subunit 2, or complex 2, is also known as succinate dehydrogenase, which was an enzyme in the Krebs cycle. And I mentioned that it was part of the electron transport chain as well. It converts, it's part of the Krebs cycle, so it has its role there to convert the succinate, but it also has its role in the electron transport chain because it reduces FAD into FADH, which is then directly used in the electron transport chain, right? NADH can come from many different sources, but for this specific subunit, succinate dehydrogenase, it comes the FADH comes directly from that. Again, it converts it directly and immediately converts the cofactor into power for pumping protons across the inner mitochondrial membrane, and that's the role of both complex 1 and complex 2. Is they use these NADH and FADH to pump protons, or H+, across the membrane from the matrix to the intermembrane space, and it's essentially like an active pump. So about this H+ gradient, like mentioned, the power source for ATP synthase is the electrical and chemical gradient between the two sides of the inner mitochondrial membrane. We want to actively pump H+ outside of the matrix into the intermembrane space. We do this by using the reduced cofactors NADH and FADH as kind of like a power source. The H+ will then go down its gradient, naturally want to come back across the membrane, and ATP synthase is actually directly tied to this process of it coming back. And we use this process of the H+ coming back to give energy or give power to the ATP synthase and convert ADP into ATP. And ADP, as you know, is the inactive form. ATP is the active form, which has energy. So as the name suggests, the electron transport chain must move electrons from complexes 1 and 2 to complex 3, complex 4, and ultimately to O2, which turns it into H2O. So, like I said, as the name suggests, electron transport chain, it's a chain that moves specifically electrons down from one place to another. Cytochrome C is one of the electron carriers that moves the electrons in between complexes. It mediates transfer from complex 3 to complex 4. Okay. Coenzyme Q is another commonly, or commonly just called Q, is another carrier molecule that mediates the transfer from complex 1 and complex 2 to complex 3. So go back to this here, this diagram, and I recommend taking pictures of all of these diagrams just so you kind of can get a visual understanding, if that helps. Complex 1 has this role, and complex 2 over here. You can see these yellow arrows from complex 1, and right here is Q, or coenzyme Q, which takes the electrons from here, moves it into complex 3, right, which does its thing. Then this cytochrome C right here will take the electrons from 3, move it right into 4. Then from 4, it'll go directly into O2 to make H2O. Okay. So these carriers are very important, and I recommend you know them, at least these two. Next is fatty, and the last topic of the day that I want to cover is called fatty acid synthesis. So this is just the general overview of fatty acid synthesis, and there's a lot to it, and there's multiple cycles. So this elongation can actually happen multiple times. This is a very long, complicated process. But for the MCAT, all you really have to know is just this, essentially. The basic idea is that we are doing what we did before, but in reverse. Instead of breaking one long free fatty acid into acetyl-CoA pieces, we are stitching together acetyl-CoA pieces to make one long free fatty acid. So before we had, say, a 16-piece fatty acid, and we broke it down into 2, 2, 2, 2, 2, 2. Now we have 2 plus 2 is 4, then add another 2, add another 2, add another 2, and then eventually we have a 16-piece fatty acid. Those will then get stitched together to form a triglyceride with a glycerol molecule and be stored as fat in our body. Okay. This process uses 2 NADH per cycle, and again, each cycle increases the chain by 2 carbons. If you're making a 16-carbon free fatty acid, you would use 7 cycles, right? And it's the same thing. Generally, the process is going from acetyl-CoA as a start point. You go into malonyl-CoA. Then you use NADPH to kind of turn this, or to add this into using an acyl carrier protein, which you don't really have to know too in depth, but you add it onto the chain, right? Essentially. And a lot of things come off. The CO2, NADH becomes NAD+, right? Water comes out, and then you just add that into the chain. And like I mentioned, this is a very long, complicated process, and this is a better, more in-depth, I would say, diagram of it if you really do want to learn it. A lot of these are very, have a lot of terminology and stuff like that in it, but this is the initiation of synthesis. Then this is the elongation cycle where it adds in, and this is where it goes in. The malonyl goes in here, and then it goes through this whole process, okay? And if you see, a lot of this is just the reverse of what we were doing, except it can't exactly go in reverse, so it has to kind of like find ways around it, okay? And like I mentioned, some of these terms like acyl carrier protein, you don't necessarily have to know all of these phosphos here and all that. So why do this? Oftentimes we eat more carbs and fat than needed in the moment, so the body decides to store them for later when it is energy starved. Therefore, it converts acetyl-CoA into free fatty acids, which are then made into triglycerides and stored away, like I mentioned. Carbs are, however, not stored in this form. Carbs are usually stored in the form of something called glycogen. So is usually what it's stored as. And the process is very similar, where it makes it into a chain that is kind of unusable in its current form, but can easily be converted back into glucose, right? But we don't really cover that in depth because, again, like this, it's not as high yield, and it's very complicated and very well regulated, right? The main thing I want you to know is the regulation. So when you're, for example, and we cover this in a different lecture, but if you're hungry, you have certain hormones that go through your body that will kind of tell your body that you're hungry and energy starved. So it'll take the fat that you have and convert it into energy to power yourself. Whereas if you're full, it'll tell your body, oh, we just had a lot of food. We don't need it right now. Turn it into fat, turn it into glycogen, turn it into whatever.